Multiple sensations and muscle movements are controlled by the nervous system. This includes sensory perceptions such as smelling flowers or touching of a hand on your shoulder, voluntary muscle movements such as controlling muscle movements to walk or talking to someone, and involuntary muscle movements such as heart beating or your stomach digesting food. Other things controlled by the nervous system include interpretation and coordination of all sensations, also regulation and coordination of voluntary and involuntary muscle movements. The nervous system is the communication and control center. The nervous system provides a means of integrating and regulating body functions through electrical signals transmitted along specialized nervous tissue called neurons. Some of the functions include the collection of information. There are receptors, which are specialized nervous system structures that monitor change in internal and external environments, also known as stimuli. Also, processing and evaluation of information. The brain and spinal cord analyze sensory information and determine the required response. There's also initiation of a response to information. Effectors is where the brain and spinal cord initiate a response. Motor information is relayed along neurons to effectors. The structural organization of the nervous system is central nervous system and peripheral nervous system. The central nervous system includes the brain and spinal cord. The brain is protected within the skull, and the spinal cord is housed within the vertebral canal. The peripheral nervous system includes nerves and ganglia. Nerves are bundles of axons of neurons, and ganglia are clusters of neuron cell bodies located along the nerves. Functional organization of the nervous system is sensory versus motor nervous system, which both have central nervous system and peripheral nervous system components. The sensory nervous system is also known as the afferent nervous system. It is responsible for receiving sensory information from receptors and transmitting it to the central nervous system. Sensory input is conveyed from receptors to the central nervous system. It is subdivided into conscious perception, also known as the somatic sensory, and unconscious, which is known as visceral sensory. The motor nervous system, also known as efferent nervous system, controls effectors and initiates and transmits motor information from the central nervous system to effectors. Motor output is transmitted from the central nervous system to effectors. Again, there are subdivisions. The conscious nervous system, also known as the somatic motor system, and the unconscious system, which is known as unconscious or involuntary control, also known as autonomic motor. Nerves are organs composed of cable-like bundles of axons, connective tissue, and blood vessels. Nerves are a component of the peripheral nervous system, or PNS. Axons bundled into structures called fascicles within the nerve. Multiple fascicles are present within the nerve, similar to the organization of skeletal muscle. The connective tissue wrappings include the epineurium, which is a thick layer of dense, irregular connective tissue, enclosing the entire nerve. It provides protection and support resembling a tough leather sheath. The perineurium is a layer of dense, irregular connective tissue wrapping around each fascicle. It provides protection and support to each bundle of axons. It also supports blood vessels. The endoneurium is a delicate layer of areolar connective tissue surrounding each axon. It separates and electrically insulates each axon. There is an extensive network of blood vessels branching through the epineurium and perineurium to become capillaries, or small blood vessels. Structural classification of nerves are based on the central nervous system component from which the nerve extends. There are cranial nerves which extend from the brain, and spinal nerves which extend from the spinal cord. Functional classification of nerves is based on the type of neuron, either sensory or motor, that a nerve contains. Sensory nerves contain neurons relaying information towards the central nervous system, and motor nerves contain neurons relaying information away from the central nervous system. Mixed nerves contain both sensory and motor neurons. Most named nerves are mixed nerves. In mixed nerves, individual sensory and motor neurons still transmit only one type of information. Ganglia are clusters of neuron cell bodies within the peripheral nervous system. It results in a swelling or an enlarged portion along the length of the nerve, often visible to the naked eye. There's the posterior or dorsal root ganglia, which is associated with sensory neurons extending into the spinal cord. Ganglia associated with motor neurons extend to autonomic effectors for the parasympathetic and sympathetic divisions. These will be discussed further in the autonomic nervous system lecture. Nervous tissue is the primary tissue of the nervous system. It is composed of two distinct cell types, neurons and glial cells. Neurons are excitable cells that initiate and transmit electrical signals. Glial cells are non-excitable cells, primarily supporting and protecting neurons. Neurons are the basic structural unit of the nervous system. Some of the special characteristics include excitability, which is the response to stimuli. Also conductivity, where electrical impulses are sent. Secretion includes the release of neurotransmitters. These are molecules that are stored in vesicles that are released and bind to excitable cells. Longevity. Most of our neurons are formed during fetal development and last into adulthood. And amitotic, most neurons lose their ability to form new cells through cell division. Neurons come in many shapes and sizes, but typically there are certain basic structural features that include a cell body, dendrites, and an axon. Dendrites are short tapering processes branching off of the cell body. Some neurons have many dendrites, while some only have one. They transmit graded potentials towards the cell body and not insulated by myelin. The axon is the single longer process emanating from the cell body. It makes contacts with other neurons, muscle cells, or glial cells. The cytoplasm is called axoplasm. The plasma membrane is called the axolemma. Axons have side branches called axon collaterals. The ends of the axon are the synaptic knob containing synaptic vesicles with neurotransmitters. The axon hillock is the triangle cone-shaped region that comes from the cell body. The cell body, or soma, houses the nucleus and cytoplasm. The nucleus contains chromatin and prominent nucleolus, which synthesize the cell body's large number of ribosomes. Pericardion is the cytoplasm within the cell body and contains the typical organelles. Chromatophilic substance is the large number of ribosomes which are attached to the endoplasmic reticulum, or free ribosomes, which readily absorb basic dyes when the nervous tissue is stained and viewed with a microscope. They appear as dark stained bodies and are referred to as chromatophilic substance. The myelin sheath insulates axons and axon collaterals. It's formed by certain types of glial cells, either neurolemocytes or oligodendrocytes. Neurofibril nodes are uninsulated regions between the myelin sheaths. The cytoskeleton is composed of microtubules, microfilaments, actin, and neurofilaments, which is a type of intermediate filament. This provides structural support and maintains neuron shape. Neurofilaments aggregate to form parallel bundles called neurofibrils. Axonal transport. Axons rely on the nucleus and organelles within the cell body for newly synthesized materials and breakdown or recycling of used materials. Substances move bidirectionally through the axon. Anterograde transport is movement from the cell body towards the synaptic knob, and retrograde transport is movement from the synaptic knob towards the cell body. Fast axonal transport is a speed at approximately 400 millimeters per day. The mechanism is movement along the microtubules. The power source comes from specialized motor proteins that split ATP to supply energy as needed. Substances can move in either antegrade or retrograde directions. Slow axonal transport is at a speed of approximately 0.1 to 3 millimeters per day. It is also known as axoplasmic flow, which is the movement that results from the flow of the axoplasm. Only anterograde from the cell body towards the synaptic body. And the substances include enzymes, cytoskeleton components, and new axoplasm for regenerating axons. Neurons vary in morphology and location. The structural classification is based on the number of neuron processes, either axons or dendrites, directly from the cell body. Multipolar neurons have multiple processes and are most common, many dendrites and a single axon. This is the most common in the human body. All motor neurons and most interneurons are multipolar. Bipolar have two processes, one dendrite and one axon. They are relatively limited in location in humans. However, some special senses, such as retina in the eye and the epithelium in the nose, are bipolar neurons. Unipolar neurons, also known as pseudo-unipolar, are single short neuron processes emerging from a cell body and branching like a T. The two processes extending fuse into one long axon. Most sensory neurons are unipolar. Anaxonic neurons have only one dendrite and no axon. They produce graded potentials, but not action potentials. Functional classifications are based on the direction in which the action potentials are propagated relative to the central nervous system. Sensory neurons, also known as afferent neurons, are neurons of the sensory nervous system. They conduct sensory input from both the somatic sensory and visceral sensory receptors. Most sensory neurons are unipolar. Motor neurons, or efferent neurons, are neurons of the motor nervous system, conducting motor output away from the central nervous system to somatic and autonomic effectors. All motor neurons are multipolar. Interneurons, or association neurons, lie entirely within the central nervous system. They receive stimulus from many other neurons. They carry out integrative functions of the nervous system, including receiving, processing, and storing information, and deciding how the body responds to stimuli. They facilitate communication between sensory and motor neurons. It is estimated that 99% of the neurons are inner neurons within our body. They are multipolar or anionic neurons. Synapses are specific locations where a neuron is functionally connected to another neuron or an effector, such as a muscle or a gland. There are chemical synapses, which are the most common in the nervous system, and electrical synapses. Chemical synapse components include a presynaptic neuron or signal producer that releases a neurotransmitter, and a postsynaptic neuron or signal receiver or target that binds to a neurotransmitter. A synapse can occur between the axon of the presynaptic neuron and any portion of the postsynaptic neuron surface, including the dendrite, cell body, or axon, except myelin-covered regions. Most commonly, a synapse is with a dendrite of a postsynaptic neuron. The synaptic knob of the presynaptic neuron does not quite make contact with the postsynaptic neuron. It is separated by a narrow, fluid-filled gap called the synaptic cleft. Neurotransmitter molecules are released from the synaptic vesicles of the presynaptic neuron into the synaptic cleft. Neurotransmitters bind to receptors on the postsynaptic neuron's plasma membrane, initiating a graded potential. Synaptic delay is associated with neurotransmitter release. This is the time between the neurotransmitter release from the presynaptic cell, its diffusion across the synaptic cleft, and the neurotransmitter binding to receptors in the postsynaptic neuron plasma membrane. Electrical synapses are less common in humans. They are composed of a presynaptic neuron and a postsynaptic neuron bound together. Gap junctions present a plasma membrane of both neurons, facilitating ion flow between cells. Cells act as though they share a plasma membrane. Electrical signals pass between cells with essentially no synaptic delay. Glial cells, also known as neuroglia, are the other distinct type of cell within the nervous system. They are found in both the central nervous system and the peripheral nervous system. They are smaller than neurons and capable of cell division. They assist neurons with various functions but do not transmit electrical signals. They provide physical protection, help nourish neurons, and provide organized supporting scaffolding for all nervous tissue. The types of glial cells in the central nervous system include astrocytes, ependymal cells, microglia, and oligodendrocytes. Astrocytes are star-like shape due to the surface projections. Their processes have contact with both neurons and blood capillaries. They are the most abundant glial cells in the central nervous system. Their functions include forming the blood-brain barrier, regulating interstitial fluid composition, and providing structural support, assisting neuronal development, altering synaptic activity, and occupying space of dying neurons. Ependymal cells are ciliated, simple cuboidal, or simple columnar epithelial cells lining internal cavities, also known as the ventricles of the brain and the central canal of the spinal cord. They have slender processes that branch extensively to make contact with other glial cells. Together with nearby blood capillaries, form choroid plexus, producing cerebrospinal fluid, and the cilia of the ependymal cell helps to circulate the cerebrospinal fluid. Microglia are small cells with slender branches and are classified as phagocytic cells of the immune system. They wander through the central nervous system and replicate in response to infection. They protect the central nervous system against organisms by engulfing and destroying them through phagocytosis. Oligodendrocytes are large cells with a bulbous body and slender cytoplasmic extensions or processes. They wrap around and insulate axons within the central nervous system to form a myelin sheath through myelination. The types of glial cells in the peripheral nervous system which help to insulate the neurons are satellite cells and neurolemocytes. Satellite cells are flattened cells arranged around neuronal cell bodies in a ganglion. Physically, they separate the cell bodies from the surrounding interstitial fluid, electrically insulate the cell body, and regulate nutrient and waste exchange. Neurolemocytes are also known as Schwann cells. They're elongated and flattened cells wrapping around and insulating axons within the peripheral nervous system to form a myelin sheath through myelination. They facilitate faster propagation of action potentials along the axon, similar to oligodendrocytes in the central nervous system. Myelination is the process by which part of an axon is wrapped with myelin. Myelin is an insulating covering around the axon consisting of repeating concentric layers of glial cells, plasma membrane. It is completed by neurolemocytes in the peripheral nervous system and oligodendrocytes in the central nervous system. The composition of myelin consists of glial cell plasma membrane. It contains a high proportion of lipids and a smaller number of proteins. The high lipid content gives the axon a distinct glossy white appearance and effectively insulates it. The process of myelination in the peripheral nervous system, the neurolemocyte starts to encircle a 1 millimeter portion of the axon. The cytoplasm and nucleus of the neurolemocyte are squeezed to the periphery as wrapping continues. Overlapping inner layers of the plasma membrane form the myelin sheath. The outer portion of the neurolemocyte, containing cytoplasm and nucleus, is called the neurolemma. Multiple neurolemocytes are required to myelinate longer axons. Gaps between myelinated portions are called neurofibril nodes or nodes of Ranvier. Myelination in the central nervous system. Oligodendrocytes in the central nervous system can myelinate a 1 millimeter portion of several axons. Cytoplasmic extensions wrap repeatedly around a portion of each axon where the myelin sheath is formed. No neurolemma is formed in the central nervous system due to the axons also being myelinated. Neurofibril nodes are also formed. Unmyelinated axons in the peripheral nervous system are associated with neurolemocytes and provide protection and support. The axon rests in a depressed portion of the neurolemma, but its plasma membrane does not form repeated layers around the axon. In the peripheral nervous system, axons are vulnerable to cuts, crushing injuries, and other trauma. Damaged axons can regenerate if the cell body remains intact and a critical amount of neurolemma remains. Regeneration depends on the amount of damage and the distance between the site of damaged axon and the structure it innervates. Neurolemocytes play an active role in regeneration. In the central nervous system, potential regeneration of damaged neurons is very limited due to oligodendrocytes do not release a nerve growth factor and actively inhibit axon growth. The number of axons crowded in the central nervous system tends to complicate regrowth, and astrocytes and connective tissue coverings can form scar tissue that obstructs regrowth. The steps involved in axon regeneration in the peripheral nervous system begins with axon severance. This occurs due to trauma. The initial response is where A portion proximal to the trauma seals off by membrane fusion and swells. The swelling is due to axoplasmic flow from the neuron cell body through the axon. The axon severed from the cell body and the myelin sheath surrounding the axon break down, which is a process called Wallerian degeneration. Debris is removed by macrophages through phagocytosis. The neurolema in the distal region survives. Formation of the regeneration tube is where the neurolema and remaining endoneurium form a regeneration tube. The axon begins to regenerate and remyelination occurs. Regeneration tube guides axon sprout, growing through it at a rate of about 2 to 5 millimeters per day. This is influenced by nerve growth factors released from the neurolemocytes. Innervation is restored as the axon reestablishes functional contact with original structures. The structure is either a receptor for sensory neurons to regain sensory perception, or an effector for motor neurons to regain control of a muscle or gland. The plasma membrane of neurons contain transport proteins responsible for moving substances across it. These include pumps and various types of channels. Pumps maintain specific concentration gradients by moving substances against a concentration gradient, requiring cellular energy. The plasma membranes of neurons contain sodium-potassium pumps and calcium pumps to maintain these gradients. The sodium-potassium pumps use a large amount of energy. Channels provide means for substances to move down or with their concentration gradient. Major types of channels in neurons include leak or passive channels, which are always open, allowing continuous diffusion of specific ions from high concentration to low concentration. Also, chemically gated channels, which are normally closed but open temporarily in response to neurotransmitter binding, allowing specific ions to diffuse across the membrane. Voltage-gated channels also normally are closed, however open temporarily in response to changes in electrical charge or potentials across the plasma membrane. Modality-gated channels are located on dendritic endings of sensory neurons and open or close in response to specific sensory stimuli like temperature, pressure, or light. These are components of sensory neurons which detect changes in the external or internal environment. A voltage-gated channel opens to permit ion movement when a specific transmembrane voltage is reached. The voltage-gated sodium channel exhibits 3 different states: resting, activation, and inactivation. Most have one gate that is either closed or open, except voltage-gated sodium channels, which have 2 gates, an activation gate and an inactivation gate, that can exhibit one of 3 states. The resting state is when the activation gate is closed, inactivation gate open, preventing the entry of sodium. The activation state is when both gates are open, allowing sodium entry, and the inactivation state where the activation gate is open, inactivation gate is temporarily closed, preventing sodium entry temporarily after activation. Some pumps and channels, which are in the plasma membrane of a neuron, are located along the entire neuron, such as sodium leak channels, potassium leak channels, and sodium-potassium pumps. Others are primarily only in one specific segment of a neuron. Neurons exhibit functional organization across 4 segments: the receptive segment, initial segment, conductive segment, and transmissive segment, each characterized by distinctive types of channels and pumps within their plasma membrane. The receptive segment includes dendrites and cell bodies, receives stimuli to excite the neuron, and contains chemically gated channels. The initial segment is located at the axon hillock and contains voltage-gated sodium channels and voltage-gated potassium channels. The conductive segment includes the axon and its terminal extensions, but not synaptic knobs. It contains voltage-gated sodium channels and voltage-gated potassium channels. The transmissive segment comprises the synaptic knobs and contains voltage-gated calcium channels and calcium pumps. The distribution of these channels and pumps aligns with the functions of each segment, facilitating the reception, initiation, conduction, and transmission of signals along the neuron. Neuron function includes the initiation and transmission of electrical currents. For this process to work, it is necessary to establish and maintain resting membrane potential that can be changed from its resting value. This is why it is important to understand the basic principles of electrical currents and Ohm's law. Electrical energy is the movement of charged particles. All usable forms of energy are available to do work. Neuron activity is dependent upon electrical energy, specifically electrical currents. Ohm's law is three important and relevant characteristics that are associated with electrical currents: voltage, current, and resistance. The relationship of these three variables is called Ohm's law. Voltage is the measure of the amount of difference in electrical charge between two areas and represents potential energy. The larger the difference in charge, the higher the voltage. It is measured in volts or millivolts. The voltage indicates the relative potential energy. Current is the movement of charged particles across the barrier that separates this difference. The greater the movement of charged particles, the greater the current. Current is measured in amps and is designated by the letter I for current intensity. Movement of charged particles, or current, is electrical energy that can be harnessed to do work. Resistance is the opposition to the movement of charged particles. This is the barrier between the charged areas. Resistance is measured in ohms. The larger the resistance, the lower the current. This description outlines how ions and the flow of charged particles contribute to electrical activity across the plasma membrane of cells. Ions like sodium and potassium carry electrical charge. These ions are distributed unevenly across the plasma membrane of cells. The unequal distribution of ions creates a difference in electrical charge on either side of the plasma membrane, resulting in a voltage difference. The phospholipid bilayer of the plasma membrane acts as resistance to the flow of ions. It typically does not allow ions to pass freely. Ion channels are proteins embedded in the membrane that can open and close, altering the resistance across the membrane. When ion channels are open, resistance decreases, allowing ions to pass through. When they are closed, resistance increases. When ion channels open, positively or negatively charged ions can flow across the membrane, generating an electrical current. This flow of ions constitutes electrical signaling within the cell and is crucial for processes like nerve impulses and muscle contraction. When the neuron is at rest, the membrane potential is more specifically called the resting membrane potential. At rest, neurons exhibit specific characteristics that contribute to their resting state and resting membrane potential. Neurons maintain concentration gradients for ions such as calcium, sodium, potassium, and chloride across their plasma membrane. These gradients are established and maintained by ion pumps and channels. The resting membrane potential refers to the voltage difference across the plasma membrane of a neuron when it is not actively transmitting signals. This potential is negative inside the cell compared to the outside. The typical resting membrane potential of a neuron is around negative 70 millivolts. Gated channels, including voltage-gated sodium and potassium channels, are closed at rest. These channels regulate the flow of ions across the membrane and play a crucial role in generating action potentials. The difference in electrical charge across the plasma membrane, known as the electrical gradient, contributes to the resting membrane potential. This gradient is mainly established by the differential distribution of ions across the membrane. At the synaptic knob, there is a concentration gradient for calcium ions. This gradient is essential for the release of neurotransmitters during the synaptic transmission. Neurons are specialized cells that transmit electrical signals. These signals move through different parts of the neuron in a step-by-step process. Dendrites are branching-like extensions from the neuron's cell body that receive signals from other neurons. These signals are typically chemical signals, known as neurotransmitters, that bind to receptors on the dendrites. The binding triggers electrical changes called graded potentials in the dendrites and cell body. These graded potentials can add up to initiate a larger signal called an action potential if they reach a certain threshold. The cell body, also known as the soma, contains the nucleus and other organelles. It integrates the graded potentials from the dendrites. If the incoming signals are strong enough and they reach that threshold, they trigger an action potential, which is a rapid and large change in electrical charge, which starts at the axon hillock. The axon hillock is where the action potential is generated. It acts as the decision point where the neuron determines whether to fire an action potential. If the graded potentials are strong enough to depolarize the membrane beyond the threshold, an action potential is initiated. The action potential travels down the axon, which is a long extension from the cell body. This is known as saltatory conduction if the axon is myelinated, meaning the action potential jumps between gaps in the myelin sheath, also known as the nodes of Ranvier. The action potential is a wave of depolarization where the inside of the neuron becomes more positive, followed by repolarization, where the neuron returns to its negative resting state. This happens as sodium ions enter the neuron and potassium ions exit. When the action potential reaches the axon terminals, it triggers the release of neurotransmitters stored in vesicles. These neurotransmitters are released into the synaptic cleft, which is the gap between the neurons, and binds to receptors on the neuron or muscle cell, continuing the process of communication. Here you can see an overview of the events that occur in each neuron segment. An action potential is a rapid electrical signal that travels along the axon of a neuron, allowing it to communicate with other cells. It happens in a series of steps involving changes in voltage across the neuron's membrane. The neuron is at rest with a stable voltage of about -70 millivolts, more negative inside the cell than outside. The resting state is maintained by the sodium-potassium pump, which pumps 3 sodium ions out of the neuron and 2 potassium ions into the neuron, creating a net negative charge inside. The neuron is ready to be activated. A stimulus, like a signal from another neuron, causes the neuron's membrane to become less negative. If the stimulus reaches a threshold of about -55 millivolts, voltage-gated sodium channels open. Sodium ions rush into the neuron, making the inside more positive. This is called depolarization. The voltage rapidly rises from -55 to +30 millivolts, creating the action potential. After reaching its peak at about 30 millivolts, the sodium channels close and voltage-gated potassium channels open. Potassium ions flow out of the neuron, making the inside of the cell more negative. This process is called repolarization. The membrane potential begins to return to its resting state. The potassium channels stay open a little too long, causing the neuron to become more negative than the resting potential, around -80 millivolts. This is called hyperpolarization or undershoot. This temporary extra negativity makes it harder for the neuron to fire another action potential immediately, creating a brief refractory period. The sodium-potassium pump gradually restores the membrane to its resting potential of -70 millivolts by pumping sodium out and potassium back in. The neuron is now ready to fire again if it receives another stimulus strong enough to reach the threshold. Graded potentials and action potentials, which are two of the types of electrical signals in neurons, are fundamental processes in neuronal communication. Graded potentials occur in the receptive segment of a neuron, including dendrites and the cell body. This results from the opening of chemically gated channels in response to neurotransmitter binding. The effect causes a local change in membrane potential, either depolarization, or more positive, or hyperpolarization, more negative, relative to the resting membrane potential. It functions to serve as a short-distance signal that can either summate or decay as they spread passively along the neuron's membrane. An action potential is generated at the axon hillock or initial segment. It's initiated by the opening of voltage-gated channels in response to threshold voltage change. Once initiated, action potential propagation or movement along the axon, which continues the conductive segment of the neuron. Action potentials are brief, rapid, all or nothing events characterized by depolarization, which is the reversal of membrane potential from negative to positive, followed by repolarization, which is the return of membrane potential from positive to negative. It serves as long-distance signals that travel actively along the axon without decay, enabling rapid communication over long distances. This is also known as nerve signals or nerve impulses. The propagation of an action potential or nerve signal along an axolemma, which is the axon plasma membrane, is influenced primarily by two factors: the diameter of the axon and the myelination of the axon. The larger the axon diameter allows for faster nerve signaling and velocity because there is less resistance to the movement of ions. The smaller axon diameter offers more resistance to ion movement, leading to slower nerve signal velocity. Myelinated axons conduct nerve signals more rapidly compared to unmyelinated axons. Myelin acts as an insulating layer around the axon, facilitating the rapid propagation of action potentials by allowing the nerve impulse to jump from one node of Ranvier to the next, also known as saltatory conduction. These factors collectively determine the speed at which action potentials travel along the axon, influencing the efficiency of neuronal communication within the nervous system. Neurotransmitters are small organic molecules synthesized by neurons. They are stored within vesicles in the synaptic knob. They are released from vesicles when a nerve signal triggers calcium entry into the synaptic knob. Their function is to bind specific receptors on target cells. This triggers a physiological response in the target cell. Some examples include acetylcholine, dopamine, serotonin, and glutamate. Neurotransmitters help to mediate communication between neurons. They influence various physiological processes such as cognition, mood regulation, and motor control. Dysregulation of neurotransmitter levels implicated in various neurological and psychiatric disorders. Graded potentials that occur in postsynaptic neurons are specifically called postsynaptic potentials. Postsynaptic potentials that result in the neuron becoming more positive or depolarized are called excitatory postsynaptic potentials. Postsynaptic potentials that result in the neuron becoming more negative or hyperpolarized are called inhibitory postsynaptic potentials. Neurotransmitters are categorized based on their chemical structure and their functional effects on target cells. There are 4 categories of neurotransmitters based on chemical structure. Acetylcholine is a unique structure compared to other neurotransmitters. It plays a critical role in neuromuscular junction and autonomic nervous system function. Biogenic amines, also known as monoamines, are derived from certain amino acids through enzymatic pathways. They are categorized into catecholamines, such as dopamine or norepinephrine and epinephrine, and indolamines, or examples are serotonin and histamine. Amino acids controversially exist regarding their dual function in protein synthesis and neurotransmission. Examples include glutamate, which is excitatory, and aspartate or serine, which are inhibitory. Neuropeptides or peptides are chains of amino acids ranging from 2 to 40. Examples include natural opiates like endorphins. Classification based on chemical function. Excitatory neurotransmitters induce depolarization of the postsynaptic membrane, making it more likely to generate an action potential. Examples include glutamate and acetylcholine at certain synapses. Inhibitory neurotransmitters induce hyperpolarization of the postsynaptic membrane, making it less likely to generate an action potential. Examples include gamma aminobutyric acid or GABA and glycine. Direct action neurotransmitters directly bind to receptors on the postsynaptic membrane, causing the opening of ion channels. This results in rapid changes in membrane potential. An example is acetylcholine binding to nicotinic receptors. Indirect action neurotransmitters bind to receptors, initiating intracellular signaling cascade via second messengers. They are slower but longer lasting effects compared to direct action. An example is neurotransmitters binding to G proteins, coupled receptors, and activating the cyclic AMP pathway. Here we're going to talk about features of neurotransmitters. Neurotransmitters are made or synthesized inside neurons. Each type of neurotransmitter is created from a specific molecule within the neuron. For example, dopamine is made from an amino acid called tyrosine. Once synthesized, neurotransmitters are stored in tiny sacs called vesicles, ready to be released. When a nerve signal reaches the end of a neuron, the synaptic terminal, it triggers the release of neurotransmitters into the synaptic cleft, which is the small gap between the 2 neurons. These neurotransmitters travel across the gap and bind to receptors on the target cell, which is either another neuron or a muscle cell. This binding can either excite or inhibit the target cell, causing it to send a signal or stop a signal, depending on the type of neurotransmitter. Once the neurotransmitter has done its job, it needs to be removed from the synaptic cleft to reset the system for the next signal. There are 3 ways this happens. Reuptake is when the neurotransmitter is taken back up into the original neuron to be reused. Enzymatic breakdown is where enzymes in the synaptic cleft break down the neurotransmitter into smaller parts, making it inactive. And lastly, diffusion. The neurotransmitter simply diffuses away from the synaptic cleft over time. Neuromodulation is a technique that changes how the nervous system works, either by stimulating it with electrical impulses or by using drugs. It helps control or adjust nerve activity, often to reduce pain or treat neurological disorders. For example, in chronic pain, a small device can be implanted to send mild electrical signals to nerves, blocking pain signals from reaching the brain. Neuronal integration and neuronal pools of the central nervous system. Neuronal circuits are various different types. There's converging circuit, diverging circuit, reverberating circuit, and parallel after-charge circuit. Converging circuits involve multiple presynaptic neurons synapsing into a single postsynaptic neuron. Its function is integration of multiple inputs to generate a coordinated response or amplify signals. An example is sensory information from different modalities such as touch, temperature, pain, converging onto a single neuron in the spinal cord for processing. Diverging circuits are a single presynaptic neuron that branches to synapse with multiple postsynaptic neurons, spreading information to different pathways. Its function is amplification and distribution of signals to multiple downstream targets. An example is motor commands from the brain diverging to activate different muscle groups involved in a coordinated movement. Reverberating circuits involve feedback loops where neurons in the circuit stimulate each other in a cyclical manner. They function to sustain or a repetitive neural activity, maintaining a prolonged response or rhythmic pattern. An example is neural circuits controlling rhythmic activities like breathing or walking. Parallel after discharge circuits are multiple presynaptic neurons that transmit input simultaneously through different pathways to converge onto a common postsynaptic cell. They function as temporal summation of signals from multiple sources to produce a prolonged or intense response in a postsynaptic neuron. An example is perception of complex sensory stimuli where multiple sensory inputs are integrated to form a unified perception.